Very good morning, ma'am. On behalf of KU College of Pharmacy, we welcome you today for this session for the QIP program. It's my proud privilege to introduce such an eminent personality to all of my delegates. Madam's biodata is a very voluminous and accomplished data which I would summarize and tell you all, because I think if I probably read the whole thing, it will take the whole section for me. So Professor Vandana Patravali is a professor of pharmaceutics at Department of Pharmaceutical Science and Technology, Institute of Chemical Technology, Mumbai. She has a rich teaching experience of over 30 years, and coming to her accomplishments, she has got a number of accomplishments in the form of awards and grants to her credit. To brief a few of her awards, she has received the Kukreja Orientation Award by the India section of International Academy of Cardiovascular Sciences in the year 2020. Dr. Manjushri Pal Memorial Award for the Best Pharmaceutical Scientist 2019. Professor Indira Parikh Women in Education Leaders Award by 7th World Education Congress 2018. Sri Amrit Modi Distinguished Researcher Award by Indian Pharmaceutical Association. She's a recipient of the Gandhian Young Technological Innovation Award 2018 under category MLC by Biotechnology Industry Research Assistance Council. She's been the Vice President of CRS-IC Control Release Society in the year 2016. She's received the OPPI Women Scientist Award in the year 2015. She's been awarded with the Dr. P.D. Patil Best Pharmaceutical Scientist of the Year Award in 2014. She's a recipient of grant awardee for her work on nano-vaccine for brucellosis using green technology, Grand Challenges Explorations by Bill and Melinda Gates Foundation. And she's got many more awards to her credits. To mention a few of the grants that she has received huge grants for her research activities. To mention a few of the organizations, she has received grants from various organizations like AICTE. She received 40 lakhs of grant received from UK India Education and Research Initiative for her work on hot melt exclusion assisted solid dispersions for oral bioenhancement of poorly bioavailable drugs. ICMR grants of 65.42 lakhs towards project entitled Rectal Microbicidal Nanotherapeutics for HIV and AIDS. ICMR grant of 18.44 lakhs for her project entitled Nanotechnology Based Diagnostic Module for Detection of Brucellosis. She's received grants from Department of Science and Technology for her projects on therapeutic approaches using controlled transdermal delivery to treat neurodegenerative diseases in aging populations. She has received grants of 58.34 lakhs from Department of Biotechnology for her work on extraction and isolation of CBUK-CON actives for developing nanocarrier-based cosmeceuticals. Another huge grant of 1 crore 79 lakhs of grant received from DBT for her project on nanotherapeutics with lipidic nanoparticles for the treatment of malaria. She has also received grants from Board of Research in Nuclear Sciences of 12.7 lakhs for her work on development and evaluation of novel brain-targeted nasal drug delivery systems. She has received 6.85 lakhs of grant from Science of Engineering Research Board for her project on development and evaluation of carrier-based novel foot and mouth disease virus vaccine for non-parental delivery. From Department of Atomic Energy, she has received grants of 4.47 lakhs for her project entitled Radiation Effects on the Controlled Release of Ophthalmic Drug Delivery Systems. And she has many more such funded projects to her accomplishments. She has several publications, national 42 and international 164. She has authored books and review articles. She has several presentations on various national and international platforms. She's an active professional member of Life Fellow, the Indian Chemical Society, member of Academic Planning and Development Committee in IFA, fellow Maharashtra Academy of Sciences, Vice President Controlled Release Society, Indian Chapter, convener of Association of Pharmaceutical Teachers of India Women Forum. She's a life member of American Association of Pharmaceutical Sciences, life member of APTI, Indian Cosmetic Technologists Association, member of Indian Society for Surface Science and Technology, life member of IPA Maharashtra branch, life member of Indian Women Scientists Association, life member of UDCT Alumni Association, and she's a patron member of Controlled Release Society, Indian Chapter. I feel humbled to have her in association for this program, and me being associated with Madam on various platforms. She's been associated with the KLE College of Pharmacy. It's a proud privilege, Madam, to have you with us today. So with this brief introduction, I hand over the session to Madam.
Thank you, Madam, for those kind words. In fact, it is my privilege to be associated with institute like KLE. And my today's talk, as you can see on the screen, is the screen visible?
Yes, ma'am.
And I'm audible enough, right?
Yes, ma'am, you're clear.
Sure. So this talk is on computational pharmaceutics role in drug targeting. So when Dr. Dandagi approached me, I had given 2-3 different topics, but probably looking at the participants, he selected this one. So it might be a repeat for maybe some of you, because this talk was recently given at Bharati Vidyapeeth in the similar lines. So this is the institute where I belong to, and Institute of Chemical Technology situated in Mumbai. And it is this institute which propels us to do better and better every day. So Madam has elaborated, I had given a very small CV, but probably she had digged out information from the website. I am really honored to be associated in this AICTE sponsored QIP, which is organized by KLE Belagavi. For those of you who are not aware of the Institute of Chemical Technology, it is a mandate for us to introduce this. This has NIRF ranking in pharmacy of 4, and it is NAC accredited with A++, highest score ever of 3.77 out of 4. And of course, we are accredited by NBA also. As far as the contents of this talk are concerned, I will be introducing this topic because it's really becoming very important. Why is it becoming important? Because now if you look at big technologies, in next 10 years, AI or the artificial intelligence is really taken up a very, very prominent role. And with this COVID situation, you have seen how we have got accustomed to use of computational tools. So same way, I'll be introducing how these can be used in pharma, how they can be used for MD, that's molecular dynamic simulations. Then I'll move on to some of the applications which we have done for developing drug delivery systems, be it usual drug delivery systems or nano carrier based drug delivery systems. So I'll be taking case studies from my own group, and then finally we will conclude. So as I mentioned earlier, now this particular era is of interdisciplinary research. No one can work in silos. You cannot work independently and give outputs which are required at the pace at which they are required. So obviously a biologist has to really communicate well with the chemist. And so with the mathematicians, it could be the colloidal scientists who have to come in picture along with the device industry. So in all, this is becoming now a real interdisciplinary situation, and all of us need to get accustomed to this. So this slide in a nutshell explains how artificial intelligence is really right now playing a role in healthcare. So wherever you see the dotted lines, they are the inputs and the solid lines are the outputs. So if you look at patients, they can put in all their data. You know that most of us have that habit. The moment any particular disease of our family members is identified, we check first with Google. What is it? What are the symptoms? What are the side effects? So you get lifestyle advice. Same way, in one of the articles, it is actually predicted that jobs, any job for that matter, which uses simple standard operating procedures, opportunities in that domain will vanish. And one of the thing mentioned is for radiologists, which is actually right now the most preferred branch. So MRI or any other images for that matter can be fed in, and computational tool will do the diagnosis for us. Physicians can feed in the unstructured data, and based on all the reports and all the data which patient has given, clinical decision can be given to physicians so they can match. And for a researcher, obviously, for researchers like us, there is lots which can be done, which you'll see in the next slides. Even the pharmaceutical industry is benefiting from this approach a lot. So this slide depicts that global artificial intelligence is expected to reach around $22,790 million by 2023. And it is growing at a very, very rapid pace, about 48.7%. That's a projection. And in fact, now I'm sure that it will cross even 50% because of the last 6-7 months, what we have seen. By geography, you can see that Asia Pacific region is going to exhibit the highest cumulative annual growth rate, which will even surpass these figures. These are the predicted values, but I'm sure it will surpass these figures now. Now, what is it and how is it that computational tools are playing a role? We can see in this particular slide that value of these is going to increase. So past was when we were using descriptive methodology, diagnostic based methodology. And future is where you can predict what is going to happen. And in a nutshell, it's a prescriptive type of a future which one will experience. So obviously, when the value increases, the complexity is going to get more and more, but you do have software engineers to come to your rescue and design programs. I'm sure all of you are familiar that there is a branch called computer-aided drug design, where one looks at the receptors, looks at the interaction a particular API will have with those receptors, and then see which one could be the best lead molecule and take it ahead to formulate and take it to the market. But now a branch which is really growing is computational pharmaceutics, of which I'm going to talk to you today. In fact, if you look at these three great scientists, Martin Karplus, Michael Levitt, and Arya Warshel, who are they? They are the Nobel Prize winners of 2013. And what do they have to say? They have said that even for chemists, computer is just as important a cheat tool as a test tube. So how can we not consider computers as an important tool? And in fact, these three scientists have got Nobel Prize in chemistry because they proposed structures of peptides which could be useful for variety of disease conditions, and predictions were done using computer-aided drug design. So this is a very, very important tool, and you can see that from 1919 to 2020, in fact, many fields are getting integrated. So decades of progress has happened. Genomic data continues to increase. You have experimental insights which are evolving. And to match with these, because we are moving from small APIs to larger molecules, computational power and speed also has to increase. So all this gets to get integrated to give the end results. If you look at the global scenario, where is it where it plays an important role? I gave you a general market scenario, but if you look even at biology, the field in which we are working, right? We come under life sciences, global market is expected to reach $12.45 billion US dollars by 2026. So in biology also, this is growing. By application, where it would be used? Preclinical drug development. I'm sure yesterday you had lecture by Dr. Chandra Mouli, and you would have seen how they've been using it for pharmacokinetics, how human body simulation software works. You would have got some idea about GastroPlus. Drug discovery and disease modeling has been going on for quite some while. Then there are cellular and biological simulations and many other fields. So it's not only being used by academia, now it is being used by industry and commercial houses too. In fact, on a regular basis, earlier genotoxicity studies were done in animals. Then came the cell lines. And now I understand from the chemical industry also that all they do is they give structures to the industry which has the know-how of the computer tools, and they can predict for them whether it would be genotoxic or not. And you know, for any API or any dosage form to come in market, it has to be non-toxic, non-genotoxic, non-mutagenic, non-carcinogenic, and so on. So one does not waste time doing all the experimental work, and it's very difficult nowadays, becoming very difficult to get permissions for use of animals. So then you have to have tools which you can use, and these tools become really very handy. If you look at the companies who are very active in this domain, you can see the name of Exleris, Centilos, and Silico Biotechnology and Nimbus Discovery, to just name a few. And obviously, this field is becoming very important because if you can see here that as biology is evolving, it is giving us newer insights. So as we get newer insights, we are asking newer and newer questions. And then comes the role of a technologist. Technologist will develop new technologies. You just heard Dr. Sivakumar from Bangalore, how newer technologies are coming into picture. So if newer technologies come, that will generate new data. And how will you process that data? You need computational tools. So obviously for that, you have to have newer softwares. And once you have this iterative and reiterative procedures, lot more new hypotheses will evolve. So this is just an example of biology. If this, you can replace it with chemistry or physics or mathematics or any of the pure sciences, and then the multiple disciplines, you would see that we are going to get into this mode. We will not have any option. So it's better that we get familiarized with all the computational tools which are available and start using them to our advantage. So this particular slide is a very important slide wherein I've indicated that one could use which softwares. For example, if you need to study ligand interactions and molecular dynamics, one could look at Schrodinger or BioSuite, Autodoc or Gold, of which Schrodinger is the most commonly used and the most preferred one. If you are looking at molecular modeling and SAR, you have Mestro, Gram, PAS, etc. People are using Material Studio nowadays very often for basic studies. In fact, in our lab, we do have both Material Studio and Schrodinger, and ICT also has Drug Discovery Studio. So we are able to use all of these to our advantage. For pharmacokinetic, GastroPlus, D+, MapCheck. And as data is getting evolved and becoming more complex, for example, when we are talking about proteomics or genomics, data analysis becomes very important. So you have GeneSpring or QSAR Pro, which can be helpful. And if you have to look at behavioral studies, because that is one area which is again going to be very, very important area. Already, you know, that Parkinsonism and Alzheimer's is on rise. Not only that, there are many psychiatric disorders happening because of whatever reasons be. So Etho Watcher is one particular program which is useful for these behavioral studies. What you can see down here are the most commonly used ones which industry is using. Now, question of interest dictates what kind of resolution you want, what kind of resolution you would use for your studies. So as you can see in this graph, it depicts the time scale, which from milliseconds is coming to picoseconds. So level of detailing increases when we start from DPD model to coarse grain to all atomic to quantum mechanism. In fact, I'm sure you all know that it's a quantum mechanics which is based on electronic motion. So it's a most evolved one. So it can give you the results in picoseconds. So obviously, level of detailing is much higher here. So as the level of detailing increases, the cost increases. You have all atomic, that is AA, wherein you look at point charges which are assigned to each and every atom. So this falls between the coarse grain model and quantum mechanism. This is electronic and this looks at all atom interactions. You have coarse grain, as I mentioned, which will look at electrostatic and hydrophobic interactions. So here, group of atoms actually, they can be clubbed together. So, and then you have DPD, which deals with, in fact, DPD stands for dissipative particle dynamics. And both DPD as well as continuum models, they depend on rheology and dynamics of the preparation. So they require a lot of time, in fact, longer time and lengths. You can see here, length scale versus length scale, which is for quantum mechanics. Armstrong range for quantum mechanics and meter range for continuum models. So that is the difference. So at present, for our pharmaceutical purposes, we are using these three depending on which particular application. But very soon, I'm sure, with the biologics coming into picture, since we have started working now in gene delivery, in proteomics, we would have to use quantum mechanics. This slide is the most important slide, I would say, because it gives you idea about atomistic MD simulations and what are the basic steps. So one starts with molecule construction. That's the first point. How will you construct a molecule? I'm sure all of you know how to use ChemDraw. But if you can go to PDB bases, as protein databases or zinc databases, most of the structures are available. So most of the structure for receptors are available. But if you are coming out with something very, very novel, obviously you may not have structures and you'll have to draw them. So you can draw them using the tools which I have earlier indicated, like Gromax or ChemDraw, etc. So you have idea about the initial geometries. Then you need to define the interatomic bonded and non-bonded forces, and that would be done using the force field. So if you know what interaction you need to see, you can decide which force fields you'll use for those particular molecules. In fact, softwares do give you some input, so that can be your basic input. From there on, you can start. Next step, these all are pre-processing step. Next step would be to set up a simulation box. What do you mean by setting up a simulation box? So just like how you take, you have a pencil box and or a geometry box and you put all your tools there, same way, this is a virtual box wherein you would be putting the molecules you want them to interact. So it could be small molecules, it could be large molecules, it could be receptors. But see that you add these molecules one by one. If you throw everything all in once, just like geometry box doesn't close, same thing will happen here. There'll be a chaos. So it is important that you go on adding your molecules one by one. And that would depend on shape and size, the boundary conditions, the molecules you are adding. So typically you start with small molecules and go on adding the other ones. Or if you're doing receptor kind of studies, you want to see, so you can add receptor first and one by one the other filling molecule. Now at this stage also there is lots happening, lot of interaction happening. So it is very important to minimize the energy. How will one minimize the energy? This is by removing the overlap which are happening between the molecules. So that means you are looking at which are the molecules which are giving similar kind of interactions. So you can minimize the energy by taking them off. And then allow your system to get simulated. So that's when your MD starts. That means allow them to react in three-dimensional space. So that is called as simulation till you get a kind of a thermodynamic equilibrium. Once that happens, you record the coordinates and or moment of atoms at predefined intervals. That means this will look at the simulation trajectory. Obviously, softwares are doing all this, but base, you need to understand base of this is Newton's law of motion, because that's what defines the trajectory, how one atom will react with other atom, or how a group of atoms will interact with other atoms. Once this is done, the post-processing step is analysis, wherein you would look at which properties you are interested in. So which property you want to look at when you're using this simulation trajectory? Is it that you want to know, like for example, in last lecture, he indicated transferosomes. So whether those transferosomes really go across the skin, is that the purpose? Or your purpose is just to know whether I'm able to solubilize my moiety in using cyclodextrin or not. So depending on the purpose, obviously you would get the analysis. So now coming on to applications of these in drug delivery system design, you can get all the physicochemical characteristics. You could know whether your systems are getting self-assembled, what is the kind of structure and dynamics of the complex which is getting formed, how the drug will interact with the system which I am proposing, whether my drug is getting loaded and how is it distributed within the system. So without doing any kind of wet lab experiments, you can know which particular ingredients are going to give me maximum drug loading in a nano system. Isn't that very important? And then you also get idea about stability, whether there's any aggregation taking place, or is it that my system is leaking so that the drug comes out in the vicinity, I'm reducing the entrapment ratio, etc. All the information you can get now virtually. You can look at interaction with the biomolecules. You can look at stability and interaction with other ions. What can happen when you are giving the drug moiety in any kind of drug delivery system and it reaches blood circulation. You know that reaching systemic circulation is most of the times our target. But after reaching to circulation, still you have seen that bioavailability of many APIs is just 20 to 40%. And there could be variety of reasons for that. So it could be maybe inhibition by enzymes, it could be the first pass effect. So all that virtually you can see, or it could be just that the moiety in question gets degraded in your pH 7.4, which is the condition of your blood plasma. It may also interact, you know, that protein binding plays a very, very important role to understand how long your drug delivery system will be there in the circulation. So you can get idea from virtual platform itself. And this already I have indicated, you can know about interaction with the membranes, whether the membrane will internalize the components which I want. It's very, very important when you're looking at active targeting. Whether you want your molecule to reach cytoplasm or it's a mitochondria where it should reach. And for gene delivery, it has to reach nucleus. So obviously, one can see that, predict that in the virtual platform, and then take them in the wet lab experiments. And intracellularly, whether the drug goes to endosomes, but does it get released or it gets automatically, you know, by macrophages gets washed off. Whether there is nuclear translocation happening, whether there's interaction with endogenous molecules or not, everything you can come to know using these computational tools. So now moving on to some of these studies, majorly when people started using the drug delivery system, scientists started using, they actually started with solubility calculations. And the other day I heard a lecture where he was talking about Florey-Huggins theory. I think BMS person was talking about Florey-Huggins theory. So what does it do? In fact, this theory again can identify, based on this theory, it can identify your API would be soluble in which particular co-former, or it could be which complexing agent, or it could be, if you're using, say, hot melt extrusion, what could be the diluent in which it would be soluble. And this is totally based on entropy. Using the same interaction parameter, you can get to know about miscibility of these ingredients too. So before doing the microemulsion pseudo-ternary diagram, you can directly actually look at the Florey-Huggins interaction parameters and then screen out This saves lot of time, lot of money, lot of energy. And that's the reason why industry is really taking or considering this particular technology nowadays. One can get idea about the glass transition temperature. There was a lecture on freeze drying in this particular QIP, and it's very important. Glass transition temperature, you have to know the TG. If you know the TG in freeze drying, you know that you have to go 20 degrees lower than that particular temperature for freezing temperature. But do you have to have the platforms? I had asked a question about accessories which are available, and very clearly the answer was given, which is the correct answer. Obviously, you do have inbuilt DSC, and you have nowadays the microscope, which can tell you at what particular temperature the freezing has to be carried out. But even without doing that, in virtual platform, you can get to know what is the glass transition temperature of the cryoprotectant or the API you are using, so that you know what could be the freezing temperature. So such small things, easily can be done once you have knowledge about these computational tools. Not only that, you know that nowadays importance in analysis is not on assay or the drug content, but it is on the impurities. Which impurities are present and whether they are within limits, because if they are not within the limits, they are the ones which are going to cause a lot of harm. So drug stability, whether it is chemically stable, maybe to the acid, base, oxidation, photo instability could be something. Everything could be judged. And obviously one can also find out partition coefficient. So going on to case study 1, wherein indomethacin miscibility in different carriers was seen. The carriers taken are PEO, polyethylene oxide, which forms the cubic delivery systems, glucose, sucrose, etc. To find out in which particular of the excipients indomethacin is miscible. So in silico correlation was done. And then final conclusion, of course, was drawn by experimental study. And in this case, they had used DSC. So what did they do? They used compass force field and looked at molecular dynamic simulation. These two things you should be familiar with. One is CED, which is nothing but cohesive energy density and refers to energy which is required to vaporize a mole of liquid per unit volume. Why CED is important? Because this is the one which is used for calculating solubility parameter. And solubility parameter delta is nothing but square root of cohesive energy density. So they looked at the compounds. You can see indomethacin, the value is 23.9. For polyethylene oxide, it's around 22, glucose 34, and sucrose 29. Now, please note that the compound or your API would be soluble or miscible in a component which has delta value very close to its own value. So from the table itself, it can be seen that PEO is very close to indomethacin. So indomethacin would be miscible in PEO. Whereas glucose is far away, 10.9 difference, and therefore it will be immiscible. Sucrose is intermittent between PEO and glucose. And then calculation of CED solubility parameters was done, and comparison with DSC analysis when it was done, they saw that because indomethacin is miscible in PEO, when you record a DSC, you don't see peak of indomethacin at its melting temperature, whereas with other two, you can see the peak. So by virtual experiment, you can know in what it is soluble or miscible. Coming to drug delivery systems, it can be applied to any kind of drug delivery system, be it lipid, polymeric, surfactant-based or amino acid-based dendrimer, newly evolving field of co-crystals, protein and peptides. Not only that, even inorganic like graphene and carbon nanotubes. So look at the lipid-based drug delivery systems. These are simplified by coarse grain model. I explained you earlier. This slide depicts actually fully atomistic, atom to atom. You can see that each atom is getting represented here. In case of B, you have united atoms. So similar kind of atoms are removed. You can see all hydrogen bonds or CH3 bonds, whatever are removed because they were similar type. So this is united atom, and coarse grain is still very coarser as the name suggests because it groups together even these particular molecules. So if you have about 150 interaction here in fully atomistic, you can simplify to united atom, there'll be about 50 interactions, and in coarse grain, there'll be only 12 interactions. So such kind of coarse grain models are very, very useful in our lipid-based drug delivery system. So this is a liposomal structure. I'm sure all of you know. I compare it with a mayonnaise sandwich. So you have, the polar groups which are there. So bread represents a polar group, and then you have in between a non-polar moiety. So your drug, of course, can be entrapped here in the non-polar yellow region, or it could be in the core itself. So how is the coarse grain model going to depict it? It is depicting hydrophobic head by blue, hydrophobic tail by yellow. So once this is done, it's a very, very appropriate approach, because you are actually capturing the essential physics of the system rather than specific chemical details. So it depends on what you are interested in. Here, if you are interested just to know what is the entrapment and how is this going to interact with the membranes in question, obviously coarse grain easily can be used. So measurable properties are with respect to the structure, the dynamics. You can look at the molecular permeation too. And this slide depicts, how the phospholipid bilayers would be seen in virtual platform. So what we just saw is represented here. You can give any color. So here I have selected the colors. So the molecular dynamic study, when you're doing the simulation study, how the stratum corneum lipid mixtures going to interact with the liposome is very well depicted here. So you can look at the liposomal structure, you can look at the skin lipid membranes, which is represented here. It could be after doing the permeation, where will my moiety get lodged? So molecular uptake can be seen. Not only that, if I'm making nanoparticles and I want to look at how they are going to interact with the cell membranes, even that can be seen virtually. You can do all the experiments of penetration enhancers and their mechanism of action virtually too. So in a nutshell, what I'm trying to tell you is this is a tool which can give you lots of information provided you understand how it is to be used. Coming to polymeric delivery, I've taken a case study wherein nanoparticles are coated with pH-sensitive polymers and their uptake is seen. Now, all this is virtual. So their uptake is being looked at, which particles would be taken in. So case A is when pH is low. When pH is low, the nanoparticles you can see are getting engulfed within no time, within 18 microseconds. But why is it happening? When pH is lower than the pKa, and we are talking about ionized polymers here, you can see that they do not coat it very rigidly. As a result, this nanoparticle is able to interact well with the membrane. Net result is that it gets engulfed. On the other hand, when pH is equal to pKa for these polymers, the monomers are coated, the polymer gets very, very strongly adsorbed. If polymer gets very strongly adsorbed onto the surface, they are bounced back from the cell membrane. You can see that end of 18 microsecond, this had got engulfed, but this is bouncing back. Whereas when pH is higher, again, you are changing the condition, pH is higher, they do get adsorbed. Meaning, unlike this case, the monomers and polymers are really getting adsorbed, but between them, the electrostatic interactions start happening. As a result, these nanoparticles are not fully covered with these polymers. So this gives them again scope to interact, and they behave, in fact, somewhere between this and this. So this particular slide really tells you which nanoparticles will get endocytosed. Obviously, it depends on the polymer which you are going to use. You should know its pH and pKa. Another case example of dendrimers. I hope you know what are dendrimers. Typically, they are made up of dendrons. The name originates from dendron, wherein you have a focal point, which is a chemically addressable group. And then you have different generation, first generation, second generation, third generation. And on these end groups, you can have any of the moieties. It could be amino acids. Typically, people are using amino acids. So you can start with surfactant also and go on developing this. Beauty of these dendrimers is that though they are looking bulky, typically they are less than 10 nanometer in size. So the generation number, as it increases, specifically for PAMAM dendrimers, which are marketed, as the generation number increases, they become more and more cationic, and because of which they become more and more toxic. So one needs to understand this very well. And our lab is working in these dendrimers, which are really very safe. And we have given one commercial formulation based on these two for topical use. So one can use the simulations to investigate interaction they are going to have with other molecules, because you are finally loading your drug molecules here. So how will you load your drug molecules if you do not know what effect the pH, salt, generation number, etc. are going to have? So all that can be done virtually. And you can go on modifying the terminal groups. This is one case study wherein PAMAM dendrimer itself was looked at which generation will behave how if it interacts with DNA. In fact, this is a very, very simplified diagram which very clearly indicates that if you have G3 dendrimer, there are minute deformations in the DNA. If you have G4, you can see that both DNA as well as the dendrimer are getting deformed. Whereas if you have G5, that's the generation number, G3, G4, and G5 are generation number of these dendrimers. So you can see here, DNA is deformed, but nothing happens to your molecule. What does this give us? What idea it gives us? So if you're looking at gene delivery, you want to send DNA or RNA or siRNA across, it should be able to destabilize the existing DNA in order to cut and replace. So virtually you can see which of my moieties, which of my generation will give me the best results, and then move ahead with the experimental part. Mechanism of endosomal escape for pH responsive dendrimers is depicted in this slide, and you can see that what you want is that nanoparticles or dendrimers, which are self-assembled systems which you have made, they should go in. That means they should go in through the cell membrane. They should get engulfed by the cell membrane. but they should be able to deliver the cargo where it is desired. So if it is gene delivery, it should deliver the gene, right? So this particular graphical abstract indicates that when the pH decreases at physiological pH, they have started interacting, but as a pH decreases, they are going through these plasma membranes inside. You can see the difference in the schematic here itself. And then when they are going in, the osmotic pressure increases so much that they rupture. It is important because if they remain like this and they'll go out of the body. But they need to release the cargo. How will they release the cargo? So as the volume increases, the osmotic pressure gets developed, the dendrimers swell. And the net result is this particular membrane comes under tension. You can see this membrane and how it is swelling and coming under tension. And if that happens, obviously you can see that the cargo would be released. And this can easily be explained by using Martini's simulation of interaction between G4 dendrimer. Dendrimer here is depicted as orange beads, and membrane, which is depicted as blue and green. So at different pH values, in the simulation box, you have to change the pH so that you can see what would happen. Case study, again from our own lab on ocular mucoretention. What have we done here? First slide, I always tell my students to prepare when they start with the basic outline of their. So if they understand this, they understand what work is expected of them and they are able to give me outline. So they understand, the student who has prepared understand the structure of eye. That means if this is cornea, cornea is lined with mucin, which is an aqueous layer where mucin is interspersed, and finally you do have a lipid layer on the outside. So this is the tear film, what you say the structure would be. Now the problem given to the student was, if I give you an antibiotic which has to be administered 3 to 4 times daily into the eye, can you convert it to once daily system? If the student has to convert it into once daily system, idea was, can she prepare drug polymer conjugates? And our hypothesis in this case was, if I make a drug polymer conjugate, whether it will get retained and closer to the cornea in the mucin. And how will I know that? If I do in silico interactions and prove that drug alone, when it is given, will get washed off, and when it is a drug polymer conjugate, it will have more interaction with the mucin. So we did all those studies, looked at the binding interactions which can happen on the ocular surface, and obviously the drug and the mucoadhesive polymer had covalent association. Idea was that we wanted, why covalent? Why we didn't want weaker bonds? Because idea was we wanted prolonged action. So when we did this particular study, what we have used is a very, very simple strategy wherein norfloxacin is actually getting reacted. That's a facile synthesis, facile because it's one pot synthesis, and with PMV MA, which is nothing but polymethylvinyl ether malic acid, and it gives you this kind of a drug polymer conjugate. And when you look at the molecular docking, we use a quick prop here. You can see that norfloxacin PMV MA, that's a drug conjugate with the polymer, has PSA score which is much, much higher than only drug. And aqueous solvation energy also for this is minus 492. So in very, very simple way, the computer simulation has told us that if you take only drug, this is the score and the solvation energy, score of surface area. But if I do molecular docking, what would happen if norfloxacin is there? How will it interact with mucin? And if I'm making a drug polymer conjugate, how will it interact with mucin? For those of you who are familiar, you would know that these red lines which are there, which I've indicated, indicate the kind of interaction the molecule is going to have. So in this case, it's only 1 or 2 red lines. As you can see, only 1, and here you have 1, 2, 3, 4, 5. So basically trying to say that this drug polymer conjugate would interact very, very well with mucin. So if it interacts with the mucin, obviously it will remain in this particular structure for longer period of time and will not get washed off. So that means it will give me a release which will be over a long time. And when we did these studies, now these are experimental studies wherein we have shown that norfloxacin from this kind of a gel matrix releases over 24 hours. And we have also looked at using the goat cornea, in fact, looked at how much muco-adhesive force would get generated. So CMC is a gold standard, and you can see that the drug conjugate which we have prepared is even better than sodium CMC. So drug as such doesn't have any mucoadhesion, but when we are making drug polymer conjugate, it has given us excellent result. The net result is such papers really do get published in very high impact journals. And the technology is so simple because it involves only one step synthesis. And if you use polymers which are regulated polymers or approved polymers, it can easily enter in the market. Taking ahead a study of polymer itself, again, this is from our lab, we looked at vaccine, and this is in fact Bill Melinda Gates sponsored project, wherein we looked at xyloglucan. You know that tamarind seeds is typically thrown, doesn't have much of use. So xyloglucan, which comes from tamarind seed, we have many patents around this. And we wanted to look at, can we make a gel using this, and that gel, can it entrap our vaccine moiety or antigen? So we have again used multifunctional and multi-responsive matrix here. Use a study on a virtual basis first to understand which of these gelators give me highest viscosity. So this is the plot. This actually is an experimental plot. What happens in presence of polyols, but this is what will give you the idea about the mechanistic evaluation. So we looked at how xyloglucan interacts with all kind of gelators. And we found that it again interacts very, very well in presence of glycerol. In fact, in presence of glycerol, you have dimeric xyloglucan at even 100 picoseconds. So interacts so very well, and therefore we took this ahead because it made our life simple, because we knew that it will have very strong HOH bonding. When do you know that it will have strong HOH bonding or hydrogen bonding? When distance is less than 2 Armstrong. So you can see the distance here, 1.01, 1.84, 1.72, etc. So this indicated that the particle which will form will actually entrap our antigen very, very well, unlike in these cases where distance is much higher. So with that we moved ahead from the vaccine. It gave us very good antibody titers. We went ahead and we had formulated it for brucellosis, went ahead. It went to actually USA for testing, and we now have results even of the infectious disease, which is a zoonotic disease, brucella. It can affect animals. The solution which we have proposed is One Health solution. It can be used for animals, but it can also be used for human beings, because the moieties or the excipients which we have selected are very, very safe. And you'll be glad to know for COVID, based on this technology, we have got a project from DBT now. So same thing, wherein I'll be just replacing the antigen using spike protein. And those studies are now ongoing for COVID. Another case study is for polyphytophenols. For those of you who are from pharmacognosy field, I have tried to keep presentation in such a way that there's some take-home message for all the people, those who are working in pharmaceutics, pharmacology, pharmacognosy, chemistry, because to me, I believe that all fields are very important and a teacher should have knowledge about all the fields. So in this case, this was a project which, as Madam introduced, was by DBT ICMR, and we were looking at, can we have some microbicide coming from this or HIV agent which comes from this study, which can be used in place of the usual antiretroviral. Why? I thought about this because all of them have a lot of side effects. And if we have something coming from the herbal source, it can go one, quickly in the market. Second, it will not be very costly. Third, if it is effective, it can be either used alone or if required, it can at least reduce the dose of the other antiretrovirals. So with this hypothesis, I screened many of the polyphenols and we've been doing a lot of work. In fact, again, I'm very glad to tell you that HIV is an envelope virus and so is COVID. So what have we done? Coronavirus. So what we have done, now we have proposed this particular study for coronavirus. And again, we have a grant there. We have already published a paper on ECGC having real potential use. And it comes from simple, green tea. And that's the reason why Ayush has indicated, for all of you to take Kada or maybe even the green tea. Black tea is also fine, not much of a difference, our studies have proved. So we started with in silico screening of all these, and these are the RMSD plots wherein you do not want standard deviation like what you can see in case of red. So your standard deviation has to be as less as possible, and you go ahead with that particular moiety, which gives you less standard deviation. Our hypothesis here, just to explain you, is that on the membrane, the cell membrane, there are CD4 receptors. And the virus has to attach through GP41 and GP120. So if these moieties have to act, they have to act as GP120 CD4 inhibitors. I was proposing them as fusion inhibitors. But I'm glad to tell you, not only they are fusion inhibitors, that means they can be used in prophylaxis. So all the sex workers or all the housewives who do not want any kind of infection, in fact, I'll say any kind because it has not only antiviral, it has antibacterial activity also, can be using this particular gel, could be a lubricant gel also. So lot of patents filed across this. And this is what in vitro screening results indicate, that yes, it does have inhibition for HIV-1, and you can see the results which we have got in 5 micromolar concentration. So 5 micromolar, meaning typically you know that you go with nanomolar concentration for synthetic moieties, but these are herbal moieties. So herbal moieties, 5 micromolar is quite good. We also looked at this particular study, we actually extended to vaginal nanomicrobicide. And again, by doing MD studies or molecular dynamic studies, we proved that THC, which is a non-colored analog of curcumin, can be used as a nanomicrobicide. So these studies were very well indicated that binding affinity for THC is much better, in fact, than curcumin. So if that happens, you can see these results. We have compared them against a gold standard, which is from BMS, Bristol Myers Squibb. And this is on a virtual mode. If it is -6 for BMS, -5. Till the time we started, it was actually BMS 806. By the time we went to in vitro screening lines, they came up with a derivative, giving a better score of -6. And look at our own moieties from our own spices, they are as good as synthetic derivatives as far as glide score is concerned. Not only glide score, in molecular dynamic studies also I have shown that these are better. So all these studies, of course, are published. So you can go through our papers if you want to know more. And this is the last study. I hope I have some time, maybe 5 minutes. Very quickly, I'll take it. Amyloid beta fibril and Alzheimer's. For this, it was a Gandhi Young Technology Award, which my student got from Health Minister and Energy Minister. So it starts with homology modeling. I'm sorry, I'm not proposing the name of this VBPN. VBP stands for my own initials. And this is student's name. So both the names have been taken together. So we started with homology modeling because 3D structure of VBPN was not available in PDB base. So we had to start actually with making that structure. So we validated, of course, using Ramchandran plot. Then we did sitemap analysis. When you do this sitemap analysis, you would know about the druggability score. The druggability is when you have score around 1. So you can see of all the various sites, one site had score which was around 1. So we went ahead with this particular site, and then we screened against various receptors, because for Alzheimer's there are a variety of receptors. But we were very, very happy that when we looked at the target selection, we wanted to look at A beta fibrils, because you know that it's amyloid beta fibrils. If it can break the plaque, it definitely will be a useful Alzheimer. So to our surprise, and you'll see in the results that we got very good results with A beta. And of course, based on the docking source as well as the docked poses, we got all the comparative results. We wanted to do very quickly animal studies, but Animal Ethical Committee did not permit. They said you do this thioflavin T binding assay first. And what is this assay? If A beta fibril is there, it interacts with thioflavin T. If it interacts, it gives off fluorescence, which is very, very, very high fluorescence. Now, hypothesis is if you have a moiety, even if this particular moiety, I can just tell you, is from a natural source. So if you have a moiety from a natural source, it will break these fibrils. If it breaks this fibrils, obviously this particular fluorescence will reduce. So we showed them, in fact, it degrades the amyloid fibrils, and then we got permission to do. We went one step ahead and said, you had asked for this assay, but we have provided you evidence by SEM. So this is how amyloid beta fibril looks under SEM. If plaque gets formed, it's a very rigid structure like this. And you can see that by adding VBPN, it has totally got deformed. It's degraded. And the concentration, this is at 2 millimolar, but we have got it in micromolar range now with formulation. So that was with pure drug. With formulation, it still goes down. So we went ahead and we formulated a microemulsion, because we were proposing that we'll give it by nasal route. Why nasal route? Because nasal route, through axonal transport, it can directly go to brain. There's no BBB involved there. So no blood-brain barrier. So that will come to our advantage. So we looked at permeation, mucoadhesion, because we wanted prolonged effect. So we formulated microemulsion, but we also formulated adhesive microemulsion. Looked at results of both. And when we did the brain targeting efficiency, you can see that the drug targeting efficiency is much higher than VBPN solutions. So if it is 445 for the solution, which also shows less of permeation, microemulsion is much, much higher. Targeting efficiency for mucoadhesive microemulsion is less, but I'm sure that this is at end of 3 hours. If I take it to 24 hours, since this has shown me higher permeation, it will also show me higher enhancement, because it will be in contact with the nasal mucosa and in contact to continuously be taken to the brain side. We looked at, because we had formulated delivery system from nose to brain, it shouldn't happen that it's taken up and the neuron starts degrading. Our whole purpose would be gone then. So you can see this is control, and this is 14 days consecutive ME we have given, but there were no signs of neuronal degradation. So histopath results were also good. So we went ahead and did pharmacodynamic studies. Here we have taken help of SKP College of Pharmacy, Dr. Milind Umekar and Dr. Tasvankar. In fact, they have helped us a lot with this particular study, wherein we have injected A beta peptide on day 0, then trained the animals for 7 days, then given whatever treatment we had to give. There's a control group also. We have taken the positive side, even donepezil, which is a gold standard, and we have given our own formulation nasally. Then again, you are training the animals. There are 2 training sessions. Then end of 28 days, of course, is brain sampling, and we have done even biochemical estimation. So they do have a stereotaxic apparatus. We also have stereotaxic apparatus, but at that particular time, our animal house was closed and no trained person available. So it was better to go to Taksande sir, who has expertise, and do this. And this is how radial maze looks. So ideally, when you are putting food item here at this end, and you're putting your animal in the middle, tendency will be to go pick up food and come. So in training sessions, that's what is taught to them. But still, it's seen that in spite of animals taken up the food from one radial arm, they again go to the same. So that is a defect. Or they go to the arm where there's no food at all. So that means they are getting confused. Their mind is getting confused. So that is a sign of Alzheimer's. So when we did these studies, we looked at working memory errors and reference memory errors. So these are related to whether they go in the same arm from where they've already had the food. That's a reference memory, or whether they go to the arm where there's no food at all. And also, what is the retrieval latency? You will be glad to see that results are very, very good. Look at the green everywhere, which is our own formulation given by nasal route. And in fact, in some cases, it is performing even better than donepezil, which is the gold standard, the drug having a lot of side effects. So now we can actually put this particular product in market for Alzheimer's. So not only that, we looked at biochemical analysis, and what is very important is brain-derived neuronal factors. Looked at 2 regions, hippocampus and frontal cortex also. And you can see the results here, that this is a group with saline, and this is with when A beta is given. So obviously when A beta is given, there's Alzheimer's. So it has to revert back. So our formulation is reverting back. It's going as close to control. So not only that, this particular formulation will have one better effect and than donepezil, that it is anti-inflammatory too. And for that, we have evaluated both TNF alpha as well as IL-6. And you can see that this is, of course, better than the control animals. So that means it is having both anti-inflammatory, because we found in literature that those, inflammation is also one of the major causes of Alzheimer's. So it will take care of inflammation. At the same time, what we have seen, and in fact, they are very excited. The SKB college is very excited to see that neurons are not only, their growth, the ones which have gone, their growth is stalled, further degeneration not taking place, but they have shown regeneration. So if regeneration happens, that means one can go back to original conditions. So in this way, using computational pharmaceutics, we started with homology modeling, did side mapping, the molecular docking, binding assay. We made a spray, that means we've gone to formulation. We've looked at it's all the characterization. Now we have finished even the stability data of 6 months. We have done permeation studies. We've done in vivo PK. PK is also completed. Now PD, to obviously results. PK is done. Histopathy is done. So all these studies have proved that this particular formulation is really very, very effective. So hopefully, next talk would be taken to clinical trials, yet to be seen. But we've got a grant from Gyati, BIRAC. We've already got a grant. The student's account gets 15 lakh rupees, not my account, not college account. Student gets directly money to take this technology further. So that is what is my motto. The conclusion is, analogous to paradigm shift in drug development in past 3 decades by computer-aided drug design, computational pharmaceutics also has great potential. So it can actually shift the paradigm of drug delivery research in the near future. These are some of the areas where we've been working very, very actively, and you would have heard probably some of my talks. And these are my research interests. We have already put medical devices in 65 countries. Many of the self-assembled systems have gone to market. Now I've shifted my focus little more to biologics, including tissue engineering, vaccines and adjuvants, as well as nanodiagnostic, and we are working a lot in these 3 areas. These are our platform technologies. Many of them, 22 of them are marketed, and these are some of the marketed products. In fact, in this lockdown period, I'm very happy to share with you that there are 4 additional products which have been launched in market, and one is expected in the next week. These are our collaborators, both from industry side as well as academia side, and the books. And this is our small lab, but each student here is a holistic scientist. And therefore, we are able to get outputs which definitely I'm really, really very proud of. We have all the facilities within the lab. So within the lab, we have 1 UV, we have 3 HPLCs, 1 FTIR, USP 1, 2, 4 apparatuses, freeze dryer, we have a spray dryer, we have Ultraterex, we have a Rotavap. I mean, you name the equipment which is required for making nanosystems, the Malvern sizer, the master sizer, as well as a Zeta sizer. Everything is available within the lab. Of course, Institute has many more facilities. And using PMG grant within the lab, I have now a cell culture lab also, so we can go a little faster. We are not dependent on others to do our cell culture work. So motto with which the lab works is Swami Vivekananda's saying, take up one idea, make that idea your life, think of it, dream of it, live on that idea. Let all the organs, the brain, muscles, nerves, every part of your body be full of that idea. Leave every other idea alone. So with this motto, our students work, because that is the way to success. Focus, focus, and focus. This is very lovely group of ours, and each one is important for me, and each one is very, very precious to me. Finally, thank you, because as Dr. Einstein said, imagination is more important than knowledge. So imagine and you can do everything. Knowledge is limited. Imagination encircles the world. So with that, I would like to thank once again Kaylee for giving me this opportunity to interact and share the work which we have done at AICTE. Thank you so much.
Thank you,
I'm
ma'am.
open to any kind of question.
Open for questions.
Thank you, Dr. Sarsija, for joining.
Oh, my pleasure. Wonderful talk, Vandana madam. Superb. Very, very informative.
I think we are privileged to have such eminent dignitaries who are witnessing all the sessions. Hello? Yeah. It's our privilege to have the dignitaries who are attending the session, all the sessions, and it's motivating us to keep us going with organizing the program. Delegates, please, you can put your questions in the comment box, or you can unmute your mics and interact with ma'am. She's very open-minded and very motivating. So if in areas of your interest, you can put up your questions.
Idea of delivering such talks is that groups come together. So be it from the same institute or from different institutes, please come together and work. And in unity is strength, that is proven time and again.
I think we are having all the comments which are very thankful to you for the session. And I think they are all motivated by your stuff, ma'am. We are all inspired and motivated to hear your sharing of your ideas on the areas of work, and definitely I hope that it has given new openings to the young researchers to come up with ideas and a strong take-home message that with a strong literature study in the area of interest and use of technology, that is the tools, simulated tools, we could reduce the time of lab work, which is more effective and which could give us reliable results. So that can be the take-home message from this session. On any front, you can always, if you are in here, you can always get in contact with ma'am, and I assure you she's always in front to give her helping hand in the areas of moving on. Thank you very much, ma'am.
Thank you so much. And in fact, thanks to Dr. Dandagi for selecting this topic. I had given him options, but thanks, Dr. Dandagi, for this topic. I hope people benefited. Thank you.
On behalf of KL College of Pharmacy, Belgavi and AICTE, thank you very much, ma'am, for being a part of this program. Thank you. So, ma'am, okay, to the participants, we would start the afternoon session at sharp 2 p.m. 2 p.m. And followed by the session, we would have a small validatory function to conclude with. Thank you.